Hello, everyone, and thank you for joining us this evening for week two of the Rugby Research and Injury Prevention Group 2026 Analytics Series. Our first intern today is Taya Maharaj. Taya, would you like to introduce yourself?
Sure. So thank you for the introduction, Brendan. My name is Taya, and I will be presenting on study types in medical research. I just sent a request to share my screen. Okay. So to begin, first let's understand what medical research is. This field helps us understand how diseases and injuries occur, how conditions are diagnosed, how treatments work, and how patient outcomes can be improved. So not all research studies are designed the same way. Different types of studies are used to answer different research questions, and together, they can provide different types of evidence. So this is relevant to us here at RRIPG because research provides the evidence needed to understand injuries, improve player safety, and develop effective injury prevention strategies in rugby. So medical research is broken down into two sections. It's called primary and secondary. So primary research involves collecting new data directly from experiments, patients, or populations. So examples of primary research include clinical trials, injury surveillance studies, surveys, and diagnostic studies. And then secondary research is basically, it does not involve collecting new data. So instead, researchers analyze and summarize findings from existing studies, and this includes literature reviews and meta-analyses. So the image here on this screen is an evidence pyramid, and it basically illustrates how different types of evidence are organized. So at the bottom of the hierarchy is individualized studies, and then more towards the top are systemic reviews and meta-analyses because they combine findings from multiple different studies and provide a broader range of available evidence. So more specifically, under the umbrella term of primary research, there's basic research, and this is usually conducted in a laboratory setting before studies are performed on humans. Examples include cell culture studies, animal studies, imaging development, and material development. So, for example, a cell culture study, you might use that to expose muscle cells to repeated stress, and this can help you understand how tissue damage occurs. Also, in animal study, you might investigate how a ligament heals after an injury. And then for material development, you might test a new helmet material before you actually use it on athletes. So, something about basic research is that it is limited in that the results that you find in laboratory conditions do not always directly translate to the human body. Also, the purpose of basic research is to understand biological processes, develop new technologies and treatments, and assess safety before moving on to human subjects. Also involved in the umbrella term of primary research is interventional studies. Another term for this is clinical trials. So what is done here is that researchers introduce an intervention and then evaluate the effects of that intervention. So examples of this include new medications, surgical procedures, rehabilitation programs, and injury prevention programs. One common example would be testing whether a new strength training program reduces the number of knee injuries in athletes. Also involved in interventional studies are clinical drug trials. So you probably hear this a lot in commercials and TV or just basic studies of science and medicine. So to go more into this very briefly, phase one of these trials basically focus on safety. Phase two focuses on the effectiveness of the drug. Phase three involves large-scale testing, and this involves randomized controls trials. And phase four involves monitoring after approval. So this image here on the right basically shows how randomized control trials are set up. And what is done here is that participants are randomly assigned to either the intervention group or the control group, and then the researchers compare outcomes between the two groups to determine the effect of the intervention. The last primary research type that I will go over is observational studies, and this involves observing participants without using intervention. So this involves the purpose of this is to identify patterns, investigate risk factors, and study outcomes. So, for example, tracking injuries during rugby season, studying whether previous injuries increase future injury risk, and using surveys and questionnaires. So, there are different types of observational studies, for example, prognostic studies, which predict future outcomes. For example, if people who typically get concussions are more likely to get concussions later on. There's also diagnostic studies, which compare a new diagnostic test with an already established method, and then case series, which describes observations from a group of patients with a similar condition. So the relevance here to RRIPG is that observational studies are used to monitor injury trends, identify risk factors, study concussion outcomes, and collect information from athletes through surveying. And then the secondary type of research under medical research involves mostly reviews and meta-analyses. So reviews are basically summarizing findings from multiple studies on a specific topic, and then meta-analyses combine data from multiple studies using statistical methods to produce a more precise estimate of an effect. So the benefits of secondary research are that there's a larger body of evidence, and you can make stronger conclusions from this, and it can help you guide clinical practice. So this image on the right here, it looks a bit complicated, but I promise you it's not. This is a forest plot and it's basically used in a lot of meta-analyses, and it basically shows the overlap in the information that is found in the different studies that the meta-analysis uses. So the relevance to RIPG with secondary research is that it helps us identify the most effective injury prevention and treatment strategies by combining evidence from many different sources. So some key takeaways, medical research involves two branches, primary and secondary research. With primary research, you generate new evidence through the use of basic research, interventional studies, and observational studies. And with secondary research, you're synthesizing existing evidence through the use of reviews and meta-analyses. Together, different study types can provide you with different types of evidence, and together they support evidence-based medicine. So these are my references that I used. And that is all. I'll now open the floor to any interns or Dr. Lopez with any questions, comments, concerns, or anything.
I have one question. What was the most interesting thing that you would say you learned, or the biggest takeaway that you learned when, putting together this presentation?
I think the biggest thing that I learned was how like different studies are, and how like when we're doing these different types of when we're trying to do a different study, you basically use a culmination of all these different types of things. And usually what I originally thought was that you would just use one primary type of study and that would guide your review or whatever it is, but you could use all different types of studies to basically generate findings.
So if you don't mind going to the first pyramid image, I think went quite fast through that. If everyone sees here, you know, this pyramid here shows the quality of the evidence, obviously, but the more important thing, it really expands. This one's probably one of the larger evidence-based pyramids I've seen that really break down each of the levels of the studies that can be done. Obviously, the systematic reviews, as Taya kind of alluded to at the end, and the clinical trials or controlled trials in between. Those are the ones in between, controlled and clinical trials. Those all fall in between the center. If she goes to, well, if you understand that the clinical drug, if you go to that slide, the clinical drug phases, right there. It shouldn't be just clinical drug trial phases. This is probably the phases for every research project kind of that you could do or implement upon populations. And it's important for you to understand, obviously the safety is the number one factor, right? When you guys start doing the IRB, start looking at different studies, you'll be able to figure out what you're going to be looking at or how you can make sure you protect your volunteers, your research volunteers, and that's really what it comes down to. And then let alone obviously effectiveness. And then when you move on to large scale testing, randomized controlled trials or large scale testing, which is like a registry, these are large scale when you can start doing it nationally, where the data starts coming in. So it's not just for drug or pharmaceutical trials. This phasing is probably for all big, large clinical trials. So just wanted everyone to understand that. Okay. Obviously you have your treatment and then your control. And whenever you do this, there's always a fine line of, who benefits more, who is exposed to more risk by not having the treatment versus having the treatment. And so even though it's easily presented here, it's not so easily implemented, because obviously you guys all can, and you can move this. You could bring that to the larger screen where everyone is on it. What happens is that you have the trials and that to look at the way the outcomes of this research is. So obviously, if some of you, work on a case study, it's at the lower end of the evidence scale, but it's still part of the population, still part of the multiple levels of the pyramid. And then as you see, you move up until the data is synthesized from multiple studies. And that's where, your data ends up in these systematic reviews, because you have particular points in your research or milestones, whatever you want to call them, that you meet expectations to include within a study. And these, variables are important to understand. I guess can look at one thing. Give me one whole second. Anyone else with any questions? When you're doing these systematic reviews, you grab the amount of papers and you take them from the different varying databases and you whittle them down from identification, screening, and then certain records excluded. And this is for a systematic review. And then you find out which articles are assessed for eligibility, why certain papers are excluded. And then finally, from those 2006 papers, you whittle it down to 18 papers that can possibly be brought into this systematic review. Then, of those 18 papers, you need to look at the data, obviously, and then, as I stated, find out if it has, and as you can see where the X's are, that's where it doesn't have data. Or if we can't freeze the top, you can't really tell how many teams the data was elucidated from. You can't really figure out the levels. And then amongst these, I'm sure you guys know, whoever knows soccer, there's multiple levels. And let alone there's no, this paper isn't on Major League Soccer, actually. And this is, like I said, basically, not all of them are Premier Leagues. So, and then what happens is that if there's limitations in the data, so say this German men's professional, right, or football league, or this German Bundesliga, the data is not, it only has severe injuries. So it actually shouldn't be included in this if it's missing certain data points, etc. Right? And then you come across, you can even see less data points that you can actually have in the synthesis or the analyses. So you first have to then pull all this data out of there, and then, and only then, can you actually create the meta analysis and a forest plot to present the data. right? The meta-analysis is one, but presenting the data, and this is where you all have to really, really kind of refine your skill sets. How do you present this data? And then how do you read the statistical significance of most of these papers, chap 2, but actually a couple of them need to be excluded. So how do you take this data, right? These large Excel files, how do you take them and then present that? So obviously, depending on what study you do, dictates how or who you're aiming to present the data to. So, I think what happened is it's interesting when you do these analyses and then how do you present the data? Are you just going to do a simple, by injury type, by position, forwards or defenders? You really have to pre-plan the entire study from the beginning to the end. And it's funny because only now are like Elon and Danielle and maybe one more of you guys are now dabbling in excuse me, the background folders of what we have of all the detail of what we've been collecting and how visionary you had to be to collect this data appropriately, and let alone to it be still viable now. So you have to kind of, when you do research, it really has to have some vision to it. You gotta have some teeth. You gotta spend the time, look at the literature as you saw with that systematic analysis, and really, really synthesize the data. Because whether you do quantitative or qualitative studies, and I know Ty, you didn't dabble too much into that, right? Yeah. It dictates how the data should be presented. There's certain standards or expectations of that. All right. I know we went off a little bit on a tangent there, but I just want to show you at the top of the pyramid, even more work goes into it, not just even collecting the data. So the analyses of it. All right. Any questions on that or for Miss Maharaj?
All right, thank you, Taya, for an amazing presentation. Our second intern today is Aaron Zeng. Aaron, would you please introduce yourself and share a screen?
Okay. Oh, hi, I'm Erin, and I'm a rising junior at Horseman. So I'm going to share my screen now. Okay, so today I will be talking about t-tests and the chi-squared test, and they're very useful statistical tests used to determine if data is statistically significant or not. So here's an overview. They are statistical tests, specifically hypothesis testing, and I'll talk about that in the next slide. And the main difference between the two is just about the type of data that they're processing. So t-tests compare the means or averages of one or two populations, and a chi-square test assesses relationships or differences between categorical data. And so t-tests are for numerical data, while chi-square tests are for categorical data. And if you look at this graphic, it helps you understand the difference between those two by giving examples. So if you look at quantitative variables, you're able to find the mean of those and use them in t-tests because you can find the means of, for example, like a list of ages, but you won't be able to find the means of a list of eye colors, for example. So onto hypothesis testing and p-value. So the goal of hypothesis testing is to take sample data to objectively determine if a claim about a population represents a true pattern or if the data that was obtained was just by random chance. So to do this process, first you have to establish a null hypothesis, which is the default assumption that there is no effect, no difference, or no relationship in a population. And then you would establish an alternative hypothesis, which is the statement about a population that proposes the specific effect, difference, or relationship being tested. So here are two examples which try to answer the question about if there's a difference between the amount of hours dogs and cats sleep. So for a null hypothesis, an example would be dogs and cats sleep the exact same number of hours, while an alternative hypothesis would be that dogs and cats sleep a different number of hours. And then this is p-value, which is assuming that the null hypothesis is true, it's the conditional probability of obtaining the test results. So here's how it works. You have the significance level, which is by standard 0.05, which is like 5%, and that's the threshold for deciding if the pattern in the data is true or most likely obtained by luck. So if the p-value is less than 0.05, then we would reject the null hypothesis because it would appear that the alternative hypothesis is statistically significant, which means that it's highly unlikely to have happened by chance, and otherwise you would fail to reject the null hypothesis. So, for example, if you got the data that dogs slept an average of 12 hours while cats slept an average of 14 hours, and you got that the p-value was 0.02, that's less than 0.05. So you would reject the null hypothesis and say that it's highly likely that the data represents a true pattern and it's not by luck that they have a difference in sleeping hours. So now I'm going to talk about t-tests, and they're used to determine if there is a significant difference between a sample mean and another value. And I'll talk more about that when I talk about the different types of t-tests. And they're usually used for a small sample group. So with the number of participants being less than or equal to 30, because otherwise you would use a z-test, and I'm not going to talk about that in this presentation. And t-tests are parametric, which means they have a normal distribution and they usually follow a normal bell-shaped curve. And they can be either one-tailed or two-tailed, which means they're directional or non-directional. So I have an example at the bottom of this slide. So if the alternative hypothesis was that dogs sleep more hours than cats, that would be directional because you're trying to see if it's statistically significant that dogs sleep more than cats, and you would only look at one end of the curve. While if the hypothesis was dogs sleep a different number of hours than cats, it doesn't matter if dogs sleep more or less than cats. It just matters if they sleep a different number. So it would be two-tailed and non-directional. So now I'm going to walk through the different types of t-tests. So first starting with the one sample t-test, it's when you would use this type of t-test when you compare the mean of a sample group to a standard value. So here's an example of a team doing 40-meter sprints, and we're looking at like their average sprint times. So the null hypothesis would be the team's average sprint time is 5 seconds. And then the alternative hypothesis would be the team's average sprint time is different from 5 seconds. So if you got the data was 6.5 seconds, there's a difference between 5 and 6.5, but the t-test would help you understand if that difference is statistically significant because you don't know if that was just by chance that they ran an average of a faster time or if it's really true that they improved and they are faster. Then you have the two samples t-test, which is also known as the independent samples t-test, and that compares the means of two distinct independent groups. So here's an example for that. You have the null hypothesis being that team A and team B have the exact same average sprint times, and then the alternative hypothesis would be team A and team B have different average sprint times. So if team A has an average of 5.2 seconds and team B has an average of 6.5 seconds, the t-test would help you understand if that was just by chance that maybe team B got lucky and they were faster, or if they truly are faster than team A. And then you have the paired samples t-test that determines whether the mean difference between pair measurements is zero or not. So usually you would use this in a case where you're looking only at one sample group, but you're looking at them at two different times, or you have another type of paired measurement. So, for example, the team average sprint times before and average before and after getting new shoes are exactly the same, or the alternative hypothesis being the team sprint times changed after getting new shoes. And if the data showed that the average improvement was negative 0.7, which is that they got faster after getting new shoes, then the t-test will tell you if um that was just by luck, that maybe like they warmed up and then they got faster, or if the shoes were actually useful. So, like we learned last week, you can use Excel to do many types of functions, and there's the t-test function. So, um, yeah, I'm not going to get too much into this, but basically, using the t-test function, you can plug in your data, and then it will give you the p-value right here. So now I'm going to talk about the chi-square test, which is used to determine if there is a significant relationship or difference between categorical data. Specifically, it analyzes frequencies and counts of categorical data, and it's non-parametric, which means it's not normally distributed and it doesn't really follow a bell curved shape like the t-test did. And there are also two types of chi-square tests. So first you have the goodness of fit test, which determines if the sample data matches a specific expected distribution. So here's an example of a player shooting 100 shots on a basketball court. So you have the null hypothesis, which is the player's shots are evenly distributed across all four quadrants. And then you have the alternative hypothesis, which is the player's shots are not actually evenly distributed. So you would have the observed data and then the expected data, which is from the null hypothesis. And then based on the chi-square test, that would tell you if it was just by chance or if it's actually true that the player's shots are not evenly distributed and it wasn't just um supposed to be evenly distributed, but it just happened not to be. And then you have the test of independence, which determines if two categorical variables are related to each other or if they're completely independent. So the null hypothesis in this example would be, it's also for basketball, if the shot outcome is independent of game location or if the shot outcome is dependent on game location. So very similarly, you would have observed data and expected data, and the chi-square test would tell you if the observed data was just by luck or if it's an actual pattern. So I don't have an Excel sheet for this, but you can also do this in Excel. So there's a specific chi-squared um, function, and then it will give you the p-value. So here's my summary. Hypothesis testing determines if data patterns are likely to be true or obtained just by random chance. And t-tests compare numerical averages between groups, while the chi-square test compares categorical counts to find relationships. And this is very relevant to RRIPG because when we do research, we're going to be working with a lot of data tables, and we can either use t-tests or chi-square tests or other statistical tests to see if our data is statistically significant. These are my references, and that's it. Thank you so much for listening.
All right, everyone. Well, we're just gonna, Aaron, that was great. Thank you so much. Um, you know, I guess I just want to kind of like break it down a little bit easier for everyone to kind of understand. And then this way, it allows you to almost like kind of envision what we're talking about. Obviously, I think when Aaron spoke about the independent, there could be paired sample t-tests, and then there's one sample t-test. You know, with the paired, obviously, you're two different times of the groups you're testing. And then with the one sample t-test, a single group against a known standard target value. Like the independent sample t-tests, two entirely separate groups. So what you're looking at is, you know, let's see how again. You know, you're looking at the comparisons of the groups in time, if that's the best way of saying it. 1, 2, and paired, right? They don't really do I guess what it is they do independent, paired, and then one sample. Those are the ones I'm used to terming. Did they actually classify them as one sample, two samples, and paired?
Yeah,
Aaron?
in the referenced articles that I found, that's how they described it.
Okay. So basically, yeah, what it is, I just don't want to get everyone confused with different, so I'll use your verbiage. One sample, two samples, and the paired samples. I think she did a good job explaining, the sprint times before and after, right? You're looking at performance, but also of a milestone with time. Okay, so that's really kind of what I wanted to show you with the t-test. And then obviously when you go to the chi-squares, you know, you're evaluating the relationships or the difference between the categorical values, as she stated. Yes, no, male, female, um, hair color, satisfaction level. So you would probably use chi-square to evaluate those types of data sets. And this is done in, surveys, market research, epidemiology, um, social behavioral sciences. They love the chi-square, right? But there's some key requirements for this chi-square. There's got to be a nominal or ordinal categories, right? And they have to be mutually exclusive independence from one category to the other category, right? We know this, right? Male and female, right? Two variable categories. And then sufficient sample size with the chi-square. I think, um, you know, you can use the t-test for smaller sample sizes, and then you can use the chi-square on larger, so you can have more validity because of the size. And then, like I think, Aaron alluded to the goodness of fit and the test of independence. So you could do those type of tests on top of that to validate your data. It's kind of interesting because I was just actually talking with um, the young doctors that we brought on the call the other day, um, and explaining to them, you need to validate the small data set you have. So you may have to do something, or you may have to do something else aside from just a t-test to validate your small cohort. And that's important for you guys to understand. All right. I don't know if I confused you guys more or maybe try to explain it a little bit. I was trying to give you where the chi-square test is applied in different cohorts. And then obviously, the requirements to get, a good chi-square analyzed. All right. On that note, like I said, thank you very much. You can unshare. Move the spotlight. Like again, the more you use the data, the more you'll feel comfortable with this. It's just like putting on a new pair of shoes. You just got to get the feel for it, when to apply these analyses. And then obviously, like I said, we're fortunate to have the group we have to look at these different data points. Any questions? I'm sorry, or on that, Aaron, we're good?
Thank you, Aaron, for an excellent presentation. Our third intern today is Jason Tay. Jason, would you like to introduce yourself and share your screen with us?
All right, thanks for the notion, Brendan. Sorry, I'm gonna share the screen real quick. Give me a sec.
Also, could you give us a short introduction of
Yeah,
yourself?
I got you. Yeah, thanks for the introduction again. My name is Jason Tay. I'm a rising junior at St. Mark's School, and I'll be presenting on sensitivity, specificity, and power, type 1 and type 2 errors. So essentially, everything's going to start here with confusion matrix. Whenever a test makes a call, reality is either going to be one way or another, and the test is either going to agree or it won't. That's going to give 2 by 2 or 4 outcomes. And so in this case, the test is going to be able to correctly identify someone who truly has condition or correctly clear someone who is truly healthy. And so these are going to be the two ways of being right, true positive and true negative respectively. The two ways of that could test be wrong are what really deserve our attention because they fail in opposite directions. One mistake is a false alarm. The test signals a problem that just isn't there. And the other is going to be a miss where the test overlooks a problem that is real. And in this case, a false alarm and a miss are not interchangeable, especially in this context, as they have different consequences. And almost everything else in this talk is going to come from treating them as just actually different. And so if we're going to collapse it all into a single score, for example, analysis, our entire nuts will just collapse as a result. And so this is going to give us two different strengths a test can have. To start, sensitivity or true positive rate on the left is essentially how well a test finds people who genuinely have condition in our context. A highly sensitive test rarely is going to miss a real case, so when it comes back negative, you can be reassured that you're healthy. Specificity, on the hand, it's a true negative rate, and so it's going to be the opposite strength, or how well a test clears people who genuinely do not have the condition. And so a high specific test rarely is going to raise a false alarm. So when it actually comes back positive, you can be able to take it seriously. The important thing to understand here is that a test is going to be rarely equally good at both, and improving one tends to come at the expense of another. If you make a test, for example, more eager to catch every real case, it's also going to start flagging more healthy people. On the other hand, if you make it more cautious about false alarms, it's going to start missing some real cases. So there is no universally best setting, it's just an optimization problem based on the context of the problem. And to move on to predictive values, so sensitivity and specificity answer one direction of the question. Given the truth, what does the test say? But in practice, professionals are going to face the reverse question. And so given a positive result, they would like to know what it actually means. And that is what predictive values give us. So a positive predictive value, or PPV, is a chance that a positive result is actually true positive. And on the other hand, negative predictive value, or NPV, is a chance that negative result is truly negative. As you can see on the right, you can see where PPV, NPV, and sensitivity, specificity lie on a table. And so these values are going to depend heavily on what's called prevalence. So the very same test is going to behave differently in, for example, different populations. Take a screening example. A test with 99% sensitivity and 99% specificity is going to sound really good, really superb. But if the condition affects a really low percentage of people, for example, only 1 in 1,000, the PPV value is going to fall to roughly just 9%. And that means more than 9% of the positives are going to be false alarms. And this is why a strong test can still make, for example, a poor screening tool, and why we always interpret a result in light of who was tested, not just how it scores on this paper. And so everything we have said so far about tests applies to most research because a study faces the same basic problem, deciding whether something real is there while you cannot be certain. Here, the two mistakes are going to get formal names. So what's called a type 1 error is a false alarm, and so concluding that an effect is real when it actually isn't. A type 2 error is going to be the miss, so failing to detect an effect that actually exists. What's worth understanding here is that we're going to just deliberately treat these two mistakes unequally. A false discovery is considered especially damaging in our case because once it's published, it can send an entire field chase something that was never there. So we hold the risk in this case of a mistake to 5%, and it relates to what Aaron's presentation before. And so it'll be significant, so it's going to be just limited to 5% conventionally. The risk of a miss, on the other hand, is guarded more loosely because it's not really fixed in advance. Instead of just having a direct cap on it, we're going to control it by designing a study that's strong enough to detect a real effect if one exists. And that particular strength has a name of its own, which is called power. So power, statistical power, is simply the chance that a study will detect a real effect when one is really present. It's just going to be the direct counterpart to the message described. So more power is going to mean fewer misses. A study with low powers, for example, can easily fail to find the very thing it's designed to find. And so you can see we have a formula here and a bunch of kind of levers we could use to adjust the power. And so we're going to start a study can gain power from a larger sample, or N, which is going to make a real effect easier to see through random noise. It can also gain power from a bigger underlying effect or an increased effect size, which is going to be the mu1 minus mu0 value you see in the formula. And it's because strong effects are easier to spot than subtle ones. It can also gain power from cleaner, less noisy measurement. So by decreasing variance/noise, which is a sigma value, a sigma variable, and it can gain power as we loosen the threshold against false alarms. So by essentially increasing the significance level or alpha. And so that's how you can manage power. And so relevance in our context, we can use it for screening tools. So in this case, sensitivity and specificity, they're going to show us whether a concussion check or, for example, movement screen or maybe return to play test is actually reliable. And we're able to catch real injuries without constantly false alarms going on. It can also help us detecting with recognition. So for example, in the case of rugby, serious injuries are going to be more uncommon than just regular, maybe less serious injuries. And so maybe even good screens, they can produce many false positives. And as a result, this can keep analysis from maybe overreacting to flags. And lastly, one of the main applications of this is small samples. So with maybe limited player pool, studies can be easily underpowered. So maybe a no difference found signal can maybe mean a weak study, not just real equivalence. Sorry. Here are my references for the talk. Thank you so much. Any questions?
No questions, anyone? Okay. Well, more importantly, Jason, great job, and not only that, but great pace, presenting a very, complicated topic. Uh, I guess, the number one thing that I would say is the sensitivity and specificity, type 1, type 2 errors. But for us, the power and the type 1 and 2 errors are kind of concerning because we already gained the sensitivity and specificity through evaluating our p-values and confidence intervals, whether or not it's relevant for that outcome, that finding. Now, what's interesting about the importance of statistical power is that it allows you to detect the true effect in the relationship. And I think, for us, we do our power analyses, evaluating the potential of what metrics we need to achieve to get, right? When we do a power analysis, we evaluate whether or not we can get to a certain threshold to arrive at those outcomes, right? So we need a certain amount of ACL injuries to understand the true outcomes within our cohort, right? Further sub-analyses would be, we need to know the amount of ACL injuries in women or ACLs in men, or if we want to break it down even further, forwards and backs or forwards and defenders, in whatever sport it is, offense, defense. So it gets the power, as long as you sustain the power analysis and statistical power, it's valid. The outcomes give you the true effect, the actual relationship. And, they're saying you're supposed to reach 80% to ensure reliability. Did you see much of that, Jason? The percentages of what they were asking for reliability or no?
Yeah, I guess like the main value I saw in the paper was around 8%. But
Yeah.
like
So that's what
that
the
was
whole, it's 0.8, right? The 80% that they're asking for sensitivity. Ideally, if you come up with anything close to it, you can say it reaches statistical power. It's close to it. You know that you're almost there. And it's almost when you're defining your discussion and your results findings. That's the importance. Anyway, I don't want to belate the point. Very good job there, son. And like I said, it's hard to kind of present all these without actually applying them. And I think, just being careful with the false negatives in the type 2, because it may actually be valid. It may actually be true versus a false negative. So just be careful with that when you're analyzing the data and your sample size. Okay? All right. Any questions on that, anyone? No. All right. Phenomenal.
Thank you, Jason, for an excellent presentation. And that will conclude this week's Rugby Research and Injury Prevention Group Analytics series. We thank everyone for joining us today.